I'm Dr. T, and welcome to my office. So today I'd like to talk a bit about transcription. So in this case, we are copying the DNA, but we are copying it into RNA. And a lot of what's going to take place is similar to that of DNA replication, but a lot simpler. A lot of the proteins just kind of simplify the whole mess. There's no back stitching. But yeah, so let's go start out. Your DNA is wrapped around proteins. Kind of the core wrap is around what we call histones, and there's other proteins associated with this. The goal here is to take this really long floppy molecule that could be very easily damaged because it's so big, and then you wrap it up and coil it nice and neatly so that it stays nice and safe. But just kind of like using your earbuds, they're wrapped nice and neatly, then you got to take them out and unwrap them in order to actually use them. So you do that with your DNA. Now, you don't unwrap the whole thing at once, you unwrap it in parts, and with the mRNA, or I should say with the RNA synthesis, because it doesn't really matter which type, they all kind of have the similar narrative. You're not unwrapping the whole thing. You unwrap part of it. Your RNA polymerase now, so instead of being a DNA polymerase, you got an RNA polymerase slides in. It's only going to read one of the two strands. The other strand is just going to sit by, kind of go on the outside, not do anything. The RNA polymerase doesn't do as much error correction, if much if any. Its goal is to make a short temporary copy. Let's just get her done. We're not going to be too fancy. If I get something wrong, well, we're not going to keep this copy around for long, and we'll just make a new copy later. So it slips into the DNA. You don't really get a fork, partially because, once again, you're only copying one side. And it's only going in one direction, so you're not trying to do the whole bubble thing. You get a little tiny bubble and it zips down. Now, in doing this, you get the RNA polymerase. There's other proteins involved called transcription factors. I'll talk about those a little bit later on when we talk about actually regulating the functions of the cells, because a lot of those are going to help get the RNA polymerase to where it needs to go. It's going to look at material on the DNA that isn't actually going to be coded, what we call a gene. It's not going to turn into a protein or anything. It's not even going to get copied except for during replication. It's kind of this what I would call metadata, what is called in cell biology circles the promoter. So this is great. I've got enough computer stuff that I think can switch between the biology and the computer, and the language of DNA is completely different, but the ideas and the needs of what is effectively an organic computer are the same as the thing behind me, down there. So instead of calling it metadata, even though it's exactly what this is, we call this the promoter region. There's actually a couple other regions. Okay, that was an aside. Back to the transcription. Okay, so we're going to copy this exactly like we would do it for DNA, but it's much, much shorter, and we're not done once we copy it. So DNA, once you copy it, there is some post-production. Some organisms will put methyl groups strategically on their DNA. That way, it's part of their error correcting. Humans will do this, but we don't do it in the way that bacteria do. And I'll talk about more of what the humans do with weird methyl groups. But some bacteria will put methyl groups on to mark which is the original and which is the copy kind of thing for their error correction. We do it for kind of a control thing, which another video. Now, that RNA. That RNA now this is going to be within the nucleus as it's on its way out. This is kind of part of it. It doesn't really get all the way out before this stuff happens. We're going to start doing some post-production. It's going to get edited, just like these videos. You see the jump cuts. Things are in the RNA that need to go away. These are going to be called introns. You've got introns and exons. I'd probably butcher that pronunciation, but you get the idea. The exons are going to be what is in the final production. The introns are the outtakes, essentially. Now, when it comes to the introns, 20, 30 years ago, we would have said, this is junk DNA. We got tons and tons of junk DNA. We probably have some DNA that's remnants of ancient viruses that's just in there for no good reason. However, a lot of the junk DNA isn't junk. It's actually used one way or another. In some cases, what you'll have is some cells will splice out some introns and not others. And by doing this, you will get different kinds of proteins. But more commonly, those introns are going to turn into small RNA molecules, which, remember, that's the oligomer form of RNA, the very generic term for it. And these can be very regulatory ones. They're going to help modify RNA. They could suppress it. That's the classic jobs of things like micro or miRNA or siRNA, depends if you're a person or a plant. These are going to help suppress RNA, so the goal is, hey, this looks like this virus, kill it! And you've just got kind of this whole suite of control functions. Additionally, you can add things to the mRNA. There's some of the small RNAs will tell proteins to come over and add it and then like put things in the middle. So you get lots of post-production with these mRNAs. Key ones, kind of as an overview, you've got the removal of introns, which may just simply get recycled right off the bat, or they may go do something useful. It just depends on that particular intron. You also are going to have a 5' cap. So on the 5' end of the mRNA, you're going to get an enzyme is going to come by, and it's going to modify that end by sticking a couple weirdly positioned nucleotides. And by doing that, that kind of, here's the start. And then once we go on the next step, this is specifically for mRNA at this point. At this step, we're going to then, as we continue on, when the ribosomes come, that 5' cap is going to help him lock on and go. If you have a tRNA, you're going to get some base modifications. tRNAs or transfer RNAs, we're going to see those guys later. But they get post-production too. Same basic story, just different set. They don't get a cap. Additionally, for the mRNA, on the other end, you're going to get what's called the poly A tail. And this is, once again, enzyme's going to come on and then just lengthen the back end of your mRNA, putting lots of adenosines on here. And what this is going to do is give a couple of things. One is kind of like a security feature. Once the mRNA goes out into the cytoplasm, or as it's migrating where it needs to go. One, it's going to form a ring. That 5' cap is going to join up with the tail. You're going to get some affiliating proteins, etc., helping you out. So first off, you got kind of a defense one. If you got a poly A tail, great. If you don't, where's your poly A tail? Are you a virus? Recycle it fast. And so, okay, so this is a way of identifying that this is a legit copy of mRNA. At least hopefully trying to defeat the viruses does not always work, obviously, because viruses are a thing. But then what it's also going to do is during the translation process where you're making the proteins from an mRNA's template, that poly A tail gets shortened as time progresses, and once it gets short enough, then all of a sudden it's like, where's your poly A tail? Yep, time to recycle it. And this gives the goal of sending a message in the form of the mRNA and then having that message self-destruct. You don't want to keep that message going indefinitely. There's always the legend of fill in the blank, and I've lost track of who gets with this legend, but fill in the blank, empress or princess or king or whatnot, looking down, seeing a flower and going, ooh, that's pretty, guard, stand over here, and then 1,000 years later, there's still a guard standing at that spot when the flower is long gone, because that order was never rescinded. So this way, it's you go to the shopping center, you buy what you need, and you don't just keep buying it without thinking, do I need it? You mark the thing off the list, and this allows you to mark this off the list. The mRNA goes away, and the cell doesn't keep manufacturing this protein ad nauseum. And this is going to help it control how much protein it has. Okay, so at this point, we've got ourselves an mRNA. It has been post-produced. Let's see if I can talk. Post-produced. It's got a poly A tail. It's got a 5' cap, and it has the introns removed. At which point, it is now time for some good old-fashioned translation. Yes. Remember, transcribing is copying, and you're going that's from the same language. So making the RNA is transcribing. Translating is going from one language to another, and that's making the protein. Because proteins are, those are amino acids, not nucleotides. Okay, with that said, have a wonderful day.